Welcome back to Bio 111. This is Chapter 8, Section 3. We're looking at light independent reactions, how light energy is used to make organic molecules. All right, summary of Calvin cycle reactions. The Calvin cycle is a series of reactions that produces carbohydrates. It's a cycle because the compound RUBP, or ribulose 1,5-bisphosphate, is reused repeatedly. Three phases of the Calvin cycle. CO2 fixation, that's where CO2, atmospheric CO2, that is CO2 from the air, is fixed. It's added to another molecule, the carbon is. CO2 reduction, and then regeneration of the RUBP. All right, those three stages. Fixation, CO2 is added to RUBP by enzyme Rubisco to generate 2,3-PGA molecules. Reduction, ADP and NADPH, are used to add an electron and make sugar, Ga3P, and 2 Ga3Ps yield 1 glucose. All right, and then regeneration of RUBP from Ga3P. All right. Zoomed in copy of the Calvin cycle. All right, let's look at this in a little bit more detail. So CO2 fixation. CO2 from the atmosphere combines with RUBP, forming a 6-carbon molecule. This reaction uses the enzyme RUBP carboxylase, or Rubisco. Rubisco is thought to be the most abundant protein on the earth. It's found in every photosynthetic organism in large quantities. All right. Each 6-carbon molecule splits into 2 3-carbon molecules, 3PG. Right. Step 2, CO2 reduction. Some of the 3PG molecules are reduced to G3P, which is the end product of photosynthesis. This uses energy and electrons supplied by ATP and NADPH produced in the light reactions, converting them back to ADP and NADP+, which returns the light reactions to be reused. And then regeneration of the RUBP. Other 3PG molecules are converted back to RUPB using energy from ADP from the light reactions. These RUBP molecules will combine with more CO2, starting the cycle again. All right. G3P can be used to create all the other organic molecules that are needed by the plant. So that's glucose, the sugar, a carbon source, of course, sucrose and starch and cellulose, larger sugars, and then fatty acids, glycerol, and amino acids. All right? So we're looking at all the building blocks, or many of the building blocks for life here. Right? Remember, amino acids, those are the subunits that make up, the monomers that make up proteins. All right, so all photosynthesizers have the metabolic pathways that are able to convert G3P to these other molecules. All right, we're going to talk about 3 different types of photosynthesis. So it happens to be that there are 3 different versions of photosynthesis, and each one is tuned a little bit better for a different environment. All right? They differ in the way that they fix CO2. Now, most of what we've been looking at is C3. So what we've been looking at the previous slides, we're talking about C3 photosynthesis. All right? It's called C3 because CO2 fixation first produces the 3-carbon molecule 3PG. It's common in plants where there's temperature and rainfall are moderate, so they're not experiencing any extreme heat. They're not experiencing any lack of water, any drought. And then photosynthesis occurs throughout the leaf. sufficient in cool, moist conditions with normal light, but hot, dry weather, when stomata closed and conserve water, CO2 cannot enter and O2 cannot leave the leaf. Remember, a stomata, we're looking at the bottom of the leaf. Stomata are these pores that can be open and closed. So little holes can be open and closed. It allows for CO2 to come in and then allows for oxygen to leave. It also allows, unintentionally, it also allows for water to escape the leaf as well. If you're in a drought condition, that's not something you want to happen. All right. The other forms of photosynthesis that we're going to see later on in this chapter provide solutions to that loss of water from the stomata being open. All right. Another problem with having to have the stomata closed a lot in the C3 plants is that rubisco is actually not that specific to CO2. It'll bind with O2. It'll bind with oxygen. And when it binds with oxygen, it's not binding with CO2, obviously, and so it's not actually doing its job. Right? So O2 will kind of poison or kill the functionality of rubisco in these plants. So it reduces the overall efficiency in these C3 plants. All right, so we've got a typical diagram of the Calvin cycle here, where CO2 is entering in here, being fixated at the 3PG, 3 carbon chain. All right, C4 photosynthesis. It's more efficient in warm and dry conditions. It's called C4 because CO2 is fixed to a 4 carbon molecule. In these plants, in C4 plants, light reactions and CO2 fixation occur in the mesophyll cells. They are then moved to a bundle sheath cells around the leaf veins, all right, where they are converted back to CO2 for the use in Calvin cycle. All right. Essentially, we are separating the light reaction from the light independent reactions such that these bundle sheath cells do not have to be in indirect contact with oxygen, right? Remember, oxygen will bind to rubisco and make it less useful. We're keeping it out of the case. So this last bullet point, O2 from the outside air does not enter the bundle sheath cells because they're surrounded by the mesophyll cells. And the bundle sheath cells there sufficient fixed CO2 and no excessive oxygen. So no competition for active sites of rubisco enzyme. So essentially, the mesophyll cells surround the bundle sheath cells and prevent oxygen from getting to rubisco in those cells. All right, so a little bit more efficient. We get rid of that O2 poisoning of rubisco in this case. So in these plants, in C4 plants, the stomata can be closed more of the time because CO2 is fixed faster and delivered to the bundle sheath cells where it can be stored for the Calvin cycle. So in warm or dry conditions, this means that the stomata don't have to be open, exposing the plant to increased risk of dehydration. And also, of course, you don't have to worry about the buildup of oxygen in these closed stomata because they're not breathing, because the bundle sheath cells are not in contact with this oxygen environment. 3% of plants species are C4. So some examples are daisies, sedges, cabbage, so that's Brassicaceae, and many grasses, so Poaceae, so that would be corn. So if you look at your left picture here, your left cartoon, we're looking at a C3 plant. We see our mesophyll cells are kind of just scattered in through the center part of this leaf. Oxygen is able to get to the bundle sheath cells easily because we have all this porous environment. The C4 plants, our mesophyll cells completely surround these bundle sheath cells. The oxygenated environment that surrounds the mesophyll cells cannot make it to the bundle sheath cells. All right. Therefore, protecting that rubisco from this oxygen poisoning. All right. And see, we have this cartoon for C4 plants, or we have this intermediate between the Calvin cycle happening in the bundle sheath cells and the light reactions happening in the mesophyll cells. And then finally, this leads us to the CAM photosynthesis. This is efficient in hot, dry conditions, such as a desert. All right. In this case, stomata are allowed to open at night. We have low temperature. It allows the plant to bring in CO2 and expel oxygen. Right? So during the night, it's bringing the CO2 and it's fixating it into an acid, and it stores that acid so that during the day, when there's light, they can close their stomata, prevent water from leaving the plant because it is a desert. It can take this acid, convert it back to CO2 for the Calvin cycle. All right? And this prevents it from being dehydrated because you have the stomata closed and you're allowed to do light reactions with the windows of the leaf closed, all the vents closed in the leaf. CAM plants include most succulent desert plants such as cactuses, agaves, and some orchids. Right, so we see an example of a CAM plant here, and that is the pineapple. That's in the same family, believe it or not, as Spanish moss, which is neither Spanish nor moss. It's native to the southeast United States, and it's a flowering plant. All right, shifting gears a little bit here. Tropical rainforest and climate change. Tropical rainforests contribute greatly to the uptake of CO2 because they are the most efficient of all terrestrial ecosystems at photosynthesis. They do it really well. They have plenty of water, all right, plenty of sunlight. All right, greenhouse gases. These are gases that are in our atmosphere that act as an insulation layer in our atmosphere and prevent thermal energy from being re-emitted back into space. So the sun heats up our atmosphere, and greenhouse gases insulate our atmosphere and allow us to retain that heat more. Right? That's very useful if you're wanting to keep it's very useful in moderation when you're wanting to keep your atmosphere a certain temperature. However, when you increase that CO2 concentration too high, it becomes a problem, right? So we have one problem we're having right now is each year clearing and burning of tropical rainforests accounts for 20 to 30% of all CO2 in the atmosphere, right? So not only are we cutting down these trees and burning them, releasing CO2, but when we cut them down, we're actually removing these carbon sinks. In other words, we remove these mechanisms that take CO2 out of the air and fixate it into things like glucose or to cellulose, the wood of the tree. All right. And some of the slash and burn technique of deforestation. All right, so we're coming on fall. We're coming on fall break. So we talk about fall colors. You look outside and you see during the springtime, during the summertime, you see green leaves, right? That is because the dominant color, the dominant pigment in those leaves are chlorophyll, A and B. Right? They're absorbing most of these other colors except for green, and they're reflecting it back. Now, chlorophyll is not very stable. It's constantly having to be recreated. During the winter times, during fall, as we approach winter, deciduous trees are adapted to let their leaves die during the winter so as to protect basically their core functions. Leaves are very sensitive to, to, to cold stress, and letting them die, taking out all the potential useful compounds from them, then just letting them die and fall off is a useful strategy for them to survive these cold periods. But during that process, while they're basically turning the leaves off and letting them die, the dominant pigment shifts from being chlorophyll to being the keratins. And so we begin to see a shift towards the orange, the yellows. So that's why we have these fall colors that we're about to see. All right. Well, that's it. That's it for chapter 8. Hope to see you again next week.